Hello, everyone, and welcome to our webinar entitled Single-Cell Multiomics, DNA Methylation and 3D Genome. I am Kenneth Day, a senior scientist here in Epigenetics group at Zymo Research, and I will be your moderator for today's event. Today's educational webinar is presented by Lab Roots and brought to you by Zymo Research. Zymo Research is known as the Epigenetics company and has specialized in products that facilitate epigenetics research for its customers for over 15 years. As our name implies, we offer a full end-to-end epigenetics approach for NGS-based profiling of chromatin, DNA methylation, and gene expression. Our workflow begins with nucleic acid extraction through bioinformatics support for our customers. Zymo remains as the gold standard for bisulfite conversion, especially for single-cell methylome analysis. The exploding number of published single-cell methylome studies show continued use of Zymo bisulfite conversion, whether by using traditional sort methods or on single-cell platforms. To learn more, please visit the link on your screen. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I now present today's speaker. Dr. Yaping Liu is an assistant professor from Cincinnati Children's Hospital Medical Center in human genetics. He earned his Ph.D. from the Epigenome Center at the University of Southern California, where he developed the innovative GnomeSeq methodology. He completed the postdoctoral training in the Kellis lab at the Broad, where he contributed to the Roadmap Epigenomics and GTEx consortia that pioneered definitions of epigenetic states across human cells and tissues. In tune with his record, his recent work unifies profiling 3D chromatin architecture by Hi-C together with bisulfite conversion to analyze methylation in the context of pads within single cells. To resolve structure at this level within single cells is a major advance for epigenetics research. Without further ado, I would like to point attendees to a more complete biography of our speaker located in the Biography tab at the top of your screen. Dr. Liu, you may now begin your presentation.
Hello, everyone. My name is Yaping Liu. It's my pleasure to present my recent work, single-cell multiomics, DNA methylation, and 3D genome. So the research goal of my lab is try to bridge the gap between genetic variation and phenotypic variations. So the focus of my lab is actually on the epigenetics side. The reason we choose epigenetics is for its definition, it's a stable, heritable phenotype resulting from changes in chromosome without alteration in DNA sequence. So basically, it has the two sides. One side is the phenotype. The other side is also has the epigenetic modification on DNA sequence itself. So it actually integrates signals from genomes, of course, the confounding issue, and as well as the environment and disease. So analyze the signal from epigenetics will tell us how the genome and the disease-like symptom or phenotype variations, how they interact with each other. So in terms of epigenetics, there's many layers in different resolutions. So for like one base pair resolutions, the cytosine will get modified by DNMT or be modified by the TAD domain, TAD family. They were demethylated. And after like about 100 base pair or KB resolutions, the DNA sequence will wrap around the nucleosomes. And in more than like KB to 25 KB resolution, you will see the loop, like the different regions of the genome will loop around each other, and functional regular elements will interact with each other. And in like 100 KB to megabasis resolution, you will see the topological associated domain. So the genome, the linear genome, will organize in the 3D genome way that will fold it into the nuclear memory. And in larger resolution, you will see the territory and compartment changes. So there's many layers and in different resolution across the epigenomes. DNA methylation and 3D genome both played a very critical role for gene regulations. Like this has been known for decades. The DNA methylation in cancers, they will silence the gene from DNA methylation promoter will silence the gene expressions, and DNA methylation will activate the genes. And also for the imprinting regions as well as the inactivation, they are all associated with, and during the development, there's two ways about the demethylations. So all these tell us like the demethylation plays a very important role for gene regulations. And very recently, the 3D genome, also more and more studies focus on the 3D genome tells us the chromatin loops also tells a lot about the gene regulation information, such as the enhancer, there will loop and activate the gene, which is about maybe 100 kb away in the downstream or upstreams. Why in this interaction maybe get disrupted in cancers. Very recently, like some groups find out the DNA methylation, which is previously people think is correlated in local scale, like correlated in just like CBG island or like 100 base pair away from each other. People find out actually DNA methylation are also correlated with each other in different resolutions. Like Johns Hopkins group find out the genetic controlled methylation clusters, and also Quinn John's lab in UCSD find out these methylation haplotype blocks. And even like for very recently in by Caspar Hansen's group in Johns Hopkins, they find out the long-range correlation discovered by 40K methylation array data. They find that some of these correlation can happen even like 100 kb away. And if you look at after you calculate the correlation of DNA methylation and then get the first principal component, you find out actually the eigenvector looks similar as high C, it's compartment A and B data. While if you are using the mean methylation level, you don't see that results. So which indicates the DNA methylation may have some of the long-range correlation. But currently, there's no existing, at that time when we published the paper, there's no existing method that can directly measure long-range interaction of epigenetic signals. Right now, of course, there's some like nanopore sequencing that you might able to get some information from that. So for those of you who are not familiar with high-C, I'll just briefly introduce what is the high-C technology. Basically, high-C can physically link the DNA from the same allele over large genomic distance. There's a few steps of the high-C. First, after you get the nuclei, we will cross-link do the cross-link. So the DNA, which is far away from each other, the proteins, they will cross-link. And then after that, you use the restrict enzyme to digest the DNA. And after that, you fill in and fill the biotin and ligate it then. So the region that is far away from each other will be ligated together. And then after you purify and digest, get rid of this reverse cross-link, and then purify and pull down by biotin to enrich these long-range interactions. You can use the tail-end sequencing to get a measure of how frequently two regions in the genome they are interacting with each other. So in like 2013, people find out since you are able to use this sequence information to measure how frequently these two regions of genome interact with each other, the genetic variation in these sequences will also be evaluated. So taking advantage of that, people are able to phase their genetics and get the phased genome by high-C sequence. And in our hypothesis is, since the genetic variation can be linked by this high-C experiment, how about the epigenetic modifications, such as the demethylation in cytosines? So this is a collaboration work with Dr. Bing Ren's group and Dr. Guoqiang Li in Bing Ren's group. So we take advantage of that. We developed this methyl high-C technology, which is combine the high-throughput, high-C, the chromosome conformation capture, and the bisulfite sequencing together. So we're able to measure two modalities at the same time in a single experiment. So the basic idea is simple, that the first few steps are similar as in situ high-C, but just after we get biotin pulled down and enrich the long-range interactions, we'll do the additional step called bisulfite conversion. And then after bisulfite conversion, we'll do the PEL-N sequence. In addition to get the 3D genome information, we're also able to get the endogenous DNA methylation information at the same time. So there's some difficult challenges for analyze this data. Think about like the high-C interaction, high-C data actually create a lot of chimeric reads. And the bisulfite degradation actually sometimes also will create a lot of chimeric reads. So this is similar as people when doing their RNA-seq. If you try to map the reads across two different exon boundary and then try to figure out where they are coming from in the genome. In RNA-seq, we are able to get the help from these exon annotations. But in high-C, actually the two regions of the genome, they may be megabases away from each other. So it's very hard to map these regions if there's a lot of chimeric reads. So if we are using using traditional DNA methylation mapping tools like Bismarck or BWA-mass, we actually get a very low mappability mapped reads. And considering that only a small proportion of these in situ high-C mapped reads is about only 30% to 40% of these reads are actually from long-range interactive reads to evaluate 3D genome. If we're using the traditional way to map the meso high-C reads, we'll get very few reads that can measure the 3D genomes. So we actually developed the computational pipeline, takes advantage of the BWA. We did some modification on that and then allow us to map the reads, the canonical reads more efficiently. Therefore, we're able to get much better mapping comparing to the traditional in situ high-C without bisulfite treatment. So we also did some like simulations, like the mapping accuracy comparing with the traditional Bismarck is similar, and the DNA methylation calling are very similar as traditional using the traditional way for mapping the reads. Okay, so after we conquer the analysis challenges, the first thing we do is try to check whether or not our assay can recapture the high-quality 3D genomes. So for those of you who are familiar with the high-C analysis, this is a typical log-10 plot in high-C analysis. So basically, we comparing the meso high-C and in situ high-C from the same mouse ES cell lines, and then we measure the distance of the fraction of the reads at a specific distance, and then we plot the distributions. The meso high-C looks like similar as in situ high-C, and we also measure the library complexity. Even after bisulfite conversion, the meso high-C library complexity looks similar as in situ high-C in different biological replicates. And we also measure the 3D genome in different resolution, from the topological associated domains resolution like 250 kb resolution to like 25 kb or 5 kb resolution, that you are able to call the loops. They are largely similar, as well as call the. So also we quantify the overlap of topological associated domain called by in situ high-C and meso high-C, as well as the chromatin loops called by Jewish pipeline. They're largely overlap with each other between two technologies in the same cell lines. So second is we try to quantify when we can recapture the high quality DNA methylation levels. So the first thing we did was we checked the CpG size we captured by these two assays. So the gold standard whole genome bisulfite sequencing from the same cell line, they capture slightly more CpG than the meso high-C. And we also checked the DNA methylation concordance at each single CpG size. They're largely similar between the two assays. So each dot here represents single CpG, and then we measure DNA methylation fraction between the whole genome bisulfite sequencing and meso high-C. The overall correlation goes up to 0.7. We look at what is the overall demethylation level between the two assays in the genome browser view that methylation in methyl high C looks similar as whole genome bisulfite sequencing and get decreases at these open completing regions such as CG island or CTCF occupied regions. So in conclusion, our assay are able to capture the 3D genome and demethylation on the same DNA moleculars. So since we're able to get the demethylation from each read, so we're able to actually, by the linked reads in two linked reads at the completing loops, the anchor 1 and anchor 2 were able to get demethylation on each read. So this is not, so taking advantage of that, this is a better example, like the imprinting gene regions, the loop anchor regions, we're able to get the demethylation status that are linked in the 3D genome from the same DNA moleculars. Therefore, we're able to calculate the correlation, demethylation correlation between these two regions by their 3D genome status. So if we look at here, if we calculate the correlation, if we just shuffle the reads here, so basically we ignore the link status defined by the high C step, that we still use the same reads in the same regions, so they have the same like average demethylation level, but then you shuffle the links between the pair reads, and then we calculate the correlations. The shuffle correlation is much lower than the demethylation that's linked by these high C reads. So this tells us how is the correlation, how is demethylation concordance at the same DNA molecular comparing that average correlation in these genomic regions. And we also measure how is that comparing with a random genomic region in genome, and how is that in the interchromosomal reads. Even in the interchromosome reads, it also shows much higher in these linked reads comparing to random shuffle reads. So we also check how is this concordance of demethylation in different compartments in 3D genome. Like compartment A usually represent the active compartment, and compartment B represent like inactive compartment. For if we look at only just linked reads without any information about whether or not there's a loop there, we see there's no significant difference between these two compartments. But if we look at the region that is called by completing loops, we see there's actually significant differences. Like we get much higher correlation in compartment A than compartment B, since in compartment A there's more gene regulatory regions links. And in different completing states, we also observe, like in these functional regions, they have a much higher concordance of demethylation comparing with the random shuffle links in the same regions. So this demethylation concordance is how we measure in each, even we get the information in the same DNA moleculars. We further ask the question, what now can measure this concordance in the single cell level, which is more which is the more nuanced way to measure this observations. So single cell demethylation profile will actually help us to get the entire demethylation in the same cells, and how is that concordant with each other in the genome-wide regions. And single cell high C is recently also provide a way you're able to calculate the 3D genome information changes in each different single cells. So right now there's a lot of single cell mono-omics technology, like single cell RNA-seq. There's tons of single cell RNA-seq technology and genomic sequence or single cell ATAC-seq for measure the accessibility and demethylation to measure the genome-wide demethylation and histone modification or 3D genomes. So why are we studying the single cell multi-omics? The single cell mono-omics will provide you separate the cells according to different levels of the epigenetic states or gene expression states, but biological system is more complicated. Sometimes the joint analysis, we hope that joint analysis of multiple modality will tell you more information and helps you to get more nuanced cell types and intermediate cellular states by using this different modality information in single cell multi-omics. So currently there's multiple technology that are able to do that, like single cell GNOME-seq able to measure the chromatin accessibility or demethylation and transcriptome. The single cell triple omics like NMT-seq or triple-seq, core-seq. They're able to measure few different omics information. And there's also a combination of RNA and proteomics or NMT or GNT to get the genome and the transcriptome information. So what we do here is we further extend our methylation technology from bulk to single cell level. So we're able to simultaneously provide these two modalities and same DNA molecular in the same single cells. So what we did is actually, after the in situ Hi-C is still in the box steps. After we sort the nuclei in 96 wells, then we do the bisulfite conversion in each well. And after we apply the post-bisulfite DNA amplification and single-strand adaptase reaction, we use a dual index to ligate and do the sequence. So the first thing we did is actually we measure, after we merge each of single cells, we want to check whether or not we can get consistent information at bulk methyl Hi-C. So we did that. We merged the cells in each single cell. So left corner is the merged single cell methyl Hi-C, and the right up corner is the bulk methyl Hi-C. We actually, after we merge these about 100 cells, single cells, we get a similar correlation pattern as bulk methyl Hi-C. And after we get the first PC, the agent vector, we get a compartment information. They show similar measurement as bulk Hi-C. We also measure the chromatin interaction frequency between the bulk Hi-C, bulk methyl Hi-C and merged single cell methyl Hi-C. And we check the log-log 10 plot. The methyl Hi-C in bulk, they're similar as we did in single cell methyl Hi-C at multi-S cell line with two different culture conditions. And in the genome-wide, it looks similar between the single cell methyl Hi-C and bulk Hi-C. So we further actually comparing with not only on the box side, we also comparing at the other, like single cell level, single cell monomics. So we first checked our consistency between the two biological replicates in single cell methyl Hi-C. The stratified correlation coefficient are very high, the 0.99 with each other, comparing with the single cell Hi-C, and which is similar level as the single cell Hi-C. We further comparing the single cell methyl Hi-C together with the single cell Hi-C in the same multi-S cell line in the same culture conditions. In a more homogeneous, like 2I culture multi-S cell, their concordance show much higher after we merge the 50 cells. And in serum, which is in the more serum culture condition, which is a more heterogeneity condition, they have less concordance, but still showing high similarity between the single cell methyl Hi-C and the single cell Hi-C. Also, we measure the demethylation levels in the serum and 2I. People already know that the demethylation level will be very different. The red dot here represents global demethylation level measured by whole genome bisulfite sequencing. They showed high concordance with the whole genome bisulfite sequencing between our methyl Hi-C, single cell methyl Hi-C and the single cell whole genome bisulfite sequencing. So next thing is, if we get the two modality information, can we use this demethylation information to cluster cell and get this the heterogeneity of cells? So what we do here is actually we sequence the multi-S cell culture in two different conditions, the serum and 2I, and then we cluster this demethylation based on one megabasis demethylation density in each cell, and then by doing the T-SNP plot, we see that serum and 2I, which is marked as red and blue, they clearly separate each other, and also the single cell methyl Hi-C can reveal the heterogeneity inside the serum cultured multi-S cells. And after we merge this demethylation in each cluster, so we get a pseudo bulk methylation level in each cluster, and then we cluster this pseudo bulk demethylation level with external demethylation data from ENCODE across different tissues. And we see that cluster 1 is, as we expected, 2I are more naive states. They are showing more more distinct from the other tissue types. And class 2 and class 3, they are clustering in different groups of the tissue types. And we further did the geo-analysis of this differential methylation region between the class 2 and class 3. That's a switch in the compartment B to compartment A level when we integrate the 3D genome information. We find that actually these get more activated regions in class 3. They are enriched in limb morphogenesis, embryonic limb morphogenesis, and limb development, which is consistent with the clustering result in panel B, that class 3 are more close with the limb and neurofacial development differentiations. And we have the one example plot that's showing in class 3 and class 2, there is a switch at HOX-D regions that switch from the class 2, the compartment B, which is a repressed state, switch to active states in class 3, compartment A. And there's there's also the differential methylation region here located at CTCF, the insulators, which indicates there's concordance demethylation changes together with the insulator changes as well as the 3D genome changes. So since we're able to get this cluster by demethylation, we further to check whether or not there's concordant changes of 3D genome there and genome-wide rather than a single loci. So we find out like classes, even with only sequence like very few cells, we merge these cells with similar random number of cells in class 1, 2, and 3. We can see that in class 2, class 3, they're very different from class 1. So since we only sequence a very few number of cells, at almost the same time, just like 1 month after our publication, Dr. Joe Acker's group, they published a paper. They used a similar technology as we did, and they did in more cells. So you can clearly see here, as we do the t-SNE of the 2 different cell types in mice, and you'll see that as you merge them, you'll see that clearly see there's a distinct the 3D genomes from these 2 clusters if you classify demethylation. And further, they apply this to the reveal heterogeneity of demethylation and 3D genome human brain. And you can clearly see here by using non-CpG methylation of CpG methylation in the neuron cells, in the brain cells, in the human brain cells, you're able to distinguish different type of neuron and different non-neuron cells. And even by using chromatin interactions, you will clearly see some of the separation at different cell types. And this is some of the example of some specific marker genes. So definitely this, in the future, joint clustering of this multi-omics with their spatial information is needed. So we're able to reveal more nuanced information here. So in summary, we developed the assay called methyl-high-C, which can combine in situ high-C with bisulfite sequencing to simultaneously capture the chromosome conformation changes and demethylation on the same DNA molecules. We are able to generate consistent information to both in situ high-C and whole genome bisulfite sequencing data from the same cell lines. And demethylation is generally concordant between 2 DNA fragments that are spatially close. And methyl-high-C can capture DNA methylation and chromatin architecture that's simultaneously in a single cell. And we're able to use single-cell methyl-high-C to discover differential chromatin conformation and demethylation in heterogeneous cells. So this project starts when I was a postdoc in Dr. Manolis Kellis' lab, and then finished when I was PI in Cincinnati Children's Hospital Medical Center. Thanks to the support from my postdoc mentor, Manolis Kellis, and our collaborator, Dr. Bing Ren and Guochang Li, who finished the experiment. And thanks for the funding support for my salary and the project from NIH, Broad, and 40 Nucleome, as well as my startup in Cincinnati Children's Hospital Medical Center. Thank you. If you have any questions, please feel free to ask me.
Thank you, Dr. Liu, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So let's get started. Our first question is, it looks like the nuclear capture rate is around 40% after proximity ligation and nuclear sorting. So when you were to look across samples, what is the rate of nuclei that meet your thresholds across samples? So for instance, if you had 250,000 reads and 10,000 contacts as your threshold, how many samples can meet that threshold?
Yeah, this is a very good question. Actually, we did, one of the reviewer of this paper also asked a similar questions. And I remember we also, I can't remember clearly now, but we show this statistic in the supplement tables, like what is the overall contacts in each cell. So generally, we get a very high quality of the library, but indeed we filter out some of the cells. We did like 296 wells, and then in the end we have about 150 cells in total for the past hour QC. So I would say, yeah, a lot of, 150 divided by like 160, 96. So there's a large fraction of cells we still keep in our analysis.
I see. Thank you for your answer. So let's go to our next question. Is it possible to predict loop anchors from concordant methylation profiles alone?
Yeah, this is a very good question. Actually, this is actually the stuff we did when we do the BARC part analysis. And we try to see whether or not we can get increase the resolution and just call the loops based on this, like demethylation. So we not only just call the loops by this demethylation concordance, we actually also use the demethylation concordance information to increase the resolution of loops calling. So to define whether or not there's a functional concordant region in these loop anchor regions. So actually, we didn't sequence deep enough. This is a power issue. If we are able to sequence higher and deeper enough, we'll get very good results. But right now, since when we do the preliminary analysis, it doesn't show. It shows in some regions we improve the accuracy, but then later since we further extended to single cell level, we decided just not incorporate that part into the final publications. Yeah.
Great, thank you. So on to our next question. Are the concordant methylation values within TADs, are these regions typically hypomethylated? In other words, what % of the contact sites are actually hypomethylated? So
you mean like the loop anchor region? How many percentage of this loop anchor region are hypomethylated?
Yeah, are they always typically hypomethylated, or do you ever see instances where they are not?
Usually they are hypomethylated, this loop, because we use a very stringent calling by Jewish pipeline to call these keycaps loops. You only get like thousands of these loops in genome Y, few thousand of these loops in genome Y. They are very conservative and high in reach. There's the CTCF contained loops and non-CTCF motif contained loops. They both show somehow their hypomethylation.
So next question. This actually kind of leads from the last question. So have you looked at transcription factor binding motifs or anything beyond just CTCF to see if there's any, like, say, cell type specific transcription factors that may help organize these loops? Or is the resolution maybe not quite good enough to see those types of things yet?
Yeah, that's a very good question. So That's also the reason we try to narrow down and increase the resolution of high-C using the demethylation concordance, further narrow down and increase the resolution, and then to check whether or not there is such a transcription factor binding site. It's definitely a very good direction to go, but at this paper we didn't mention that.
Great, thank you for your answer. One more question. So when you use the single-cell methyl high-C method, particularly in your presentation, you've shown that it looks at, you can look at cellular differentiation effects, like during embryogenesis or looking at differentiation of embryonic stem cells. Have you tried to look at, say, somatic cells from adult tissues to see if this could be used for looking at, say, disease states in adult tissue?
Yeah, definitely. That's the I believe the next step both of Bing's lab and my lab were doing.
So when you look at the CPGs in your single-cell methyl high-C, what percent are those CPGs in common across the captured nuclei?
Yeah, it's very sparse. I have to admit it. This is the problem in like single-cell demethylation. It's similar as the single-cell whole genome bisulfite sequencing.
Yeah.
The data is sparse, so if you're doing like single CPG size comparison across the cells is really challenging. So without imputation, it's really challenging. That's why the main purpose of any analysis we show here is actually we use the 1 MAC basis, non-overlap window, and then do the clustering. And some groups like the Duracker group, they use the 100 kb non-overlap window to do that. So that might get these problems. But indeed, that's a challenging part.
That's a challenge, yeah. So this is more of a technical question. We noticed in one of your recent publication that you used 2 different bisulfite kits from Zymo Research, and we're kind of curious. What was your purpose in using 2 different kits?
Oh, you mean like in our publication, the bark part we use gold, and in the single cell part we use lightning, right?
Yeah, I think you use gold and direct.
And direct, sorry. Yeah, direct. Yeah, this is just.
That's okay.
Yeah, it's just at a time. This is actually also I asked Guo Qiang. Actually, he will be a better person to answer this question. He told me like at that time they just happened to have a gold kit there, so he tried that. But then later in the gold, because the gold kit is good, it's okay for bulk level, but for single cell level, the input is even lower, and we want to we actually didn't. So later he switched to the direct Zymo kit. Yeah.
I see.
Yeah.
Thank you for your answer. So I think we may have time for one more question. This is more of a kind of a global question. What is the future, you believe, for single-cell methyl high-C? We've touched on it a little bit. And what are some of the research questions or projects that you believe this method will make an impact?
Yeah. So this is a very general question. It's also one of the direction we are going to do is, so definitely there's few directions to go. Like one is you can increase the modality, get an other additional modality involved in experiment, and in terms of experiment side or in terms of the analysis side, as I mentioned in the presentation, like joint clustering of cells, including their spatial information, is challenging. So definitely it's worth to go. And in terms of their applications, so I think Joaquin's group and Dr. Binren's group are doing that right now is extend this application to different tissue or cell types. There's a lot of heterogeneity tissues such as cancers or brains, which Joaquin already did in prefrontal cortex, and there's also many other tissue types. So there's a lot of applications, and the problem is right now our understanding about the 3D genome heterogeneity in different cells is limited. So for the heterogeneity in demethylation, people already noticed that in the last few years, like since 2014, in the single cell demethylation, single cell whole genome bisulfite sequencing was invented, people look at different cell types and find out there's heterogeneity in different, like Joanna's group, the pioneer work find out in different layers of the neuron, they show in different demethylation. So maybe in future, when we know more information, more function about 3D genome changes, we'll get more advanced application for that we are able to simultaneously capture this interaction between the two layers, and of course, many other layers.
Great, thank you so much for your answer. Thank you again, Dr. Yaping Liu, for your time today and for presenting your very important research. We would also like to thank LabRoots and our sponsor, Zymo Research, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of your registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thanks for attending and goodbye.